That's honestly how I felt when I was working in the industry. But then I like interacting with my coworkers, it was like, "Oh, you guys are all the same. I don't feel so bad." No one knows anything and no one understands anything because everything is so confusing all the time. Oh man. Thank you. I'm gonna grab these headphones to see if you're the best. Amy's the best. Um Thank you, thank you. I thought it was appropriate. I thought it was appropriate. It it was fun to do first because I think this is going to be a lot more difficult than the other subjects that I'm doing. Like vitamin D is going to be a cakewalk compared to this. Transferring liquid from tube to tube, that's what we went to school for. It's like, and then you feel like it's what is it? It's like hard mode when you have like the multi-channel pipettes. Oh my god. Oh man, I used to do so many conjugations. So many conjugations. I do miss it a little bit. I miss purifying antibodies. I miss purifying what was it? I think it was like donkey anti-goat antibodies would be in like the really big columns and I carried around like it was my baby. So, yeah. All right. I am like sufficiently pumped. Um Oh my god. There should be. There should be a an achievement for dual wielding pipettes. It really like uh it at my other job when I was in academia, it was a lot of pipetting and a lot of like little tubes, having to open and close little tubes. And this guy that they sent to work for me, he couldn't open his hands, okay? He processed so many samples and did this and this so many times that he couldn't open his fucking hands. Awesome. Thank you, Amy. Yeah, I mean, you got it sometimes. All was subcryptic. Okay, this is a frickin' party, so I am I am I am eager to get into this. Plot twist. Dopamine is my favorite molecule, of course, but I think it's really cool as a reward signal, you know, like telling you that you like something and stuff like that, but I I was always a little more fascinated for whatever reason by the fact that like it has like these motor functions so that you can like I guess the point would be like go get that reward you want. Like I thought that that was so fascinating. So there are a lot of reasons why dopamine is like my my favorite guy. Yes. Oh, I agree. I agree. Yeah, the Rainin. There's no other one in my opinion. Dopamine is so gender for her. Oh my god. Dopamine I feel like I don't know if you guys will agree with this, but I feel like it's one of the most well-known molecules. Like I mean, okay. Here we go. The I don't see memes about nearly as many other molecules than I do about dopamine. Overwhelmingly. If there's going to be a meme about a particular scientific thing, it's going to be about dopamine. Uh and I am obsessed with that. I mean, everyone understands that like dopamine kind of equates things that you like. Um and so a lot of these reductive takes on it I find really legitimately entertaining. Um and this one in the middle actually the where is it? This guy, yeah, this one here. I found this on a blog and the name of the blog was dopamine is not the reward signal and I was like, "Really?" And I clicked on it and it was they were just complaining about dopamine memes because oh my god, like people just like don't get it. Like they don't even know what they're like talking about. And what did they say? Literally in like the first part of the blurb, oh, it said Okay, so dopamine isn't just your reward chemical. Like, come on, get off your high horse, motherfucker. Literal clickbait. Yeah, literally. Like, dopamine memes are funny, and memes are also supposed to be really, really reductive and watered down. I would not want my memes to be anything besides that. Um, I only understand if you are like an expert on things that are really complicated, then you're always going to be like, oh my God, it's so much deeper than that. Oh my God, no one understands me. And I kind of do understand that. This molecule will surprise you. Yeah, so I don't know. Watering it down is fine, but I'm here to spike those waters. We're gonna spike it up because we're gonna go a lot deeper than the memes will ever be able to take us, and it's gonna be really, really exciting because where's the one? Technically, the only two things you enjoy, serotonin and dopamine. Like, you know, I would go so far as to say dopamine. You don't like dopamine, actually. Dopamine just fucking tells you what you like. It fucking puts a gun to your head and is like, you like this. This is the thing that you like. I'm giving you're gonna exhibit approach behavior to this. So it's more complicated, but it's even more interesting than you could ever imagine. So why don't we get some dope knowledge going? This this is a summary of like the big ticket functions that dopamine has. I'm gonna be mainly focusing on the functions that take place in the central nervous system because the ones that are peripheral, they're really interesting. I mean, dopamine I think increases salt excretion as a way to help like lower your blood pressure, so that's really cool. But um, those things are not as well studied, and so I can't really like explain them as well as I can the stuff in the central nervous system, which is funny because usually that stuff is batshit. But I am definitely gonna go over reward, and you can bet your booty I'm also gonna bring up this and this and this and definitely this. I mean, we're gonna go the whole 9 yards with dopamine's actions in the brain. Um, and I am not I'm not a neuroscientist or a neuroanatomist or a psychologist. Like, I deal with the nitty gritty itty bitty aspects of this physiology, so uh, that's why I'm mainly going to be focusing on those things, and it's why the first thing we're going to do is zoom in. Zoom in. Loop of Henle. Oh my god, not actually in the loop of Henle. Oh my god, it might. It's after the loop of Henle, actually, in the distal convoluted tubule and the collecting duct. Um, that's actually where like a ton of other substances like aldosterone and I think ADH go in and sort of like do their thing. Um, the loop of Henle, I believe, is reserved for like impassable. Nothing goes in or out. I am impenetrable. I'll have to check up on that. Yeah, super surprising. Like I didn't know that dopamine had these peripheral functions at all. In the pancreas too. Apparently it affects insulin. Again, no one really, I don't know, like we don't really study that as much, so I can't really like explain them, but it's just like, hey, there's insulin or there's dopamine receptors in the pancreas. Well, what's the big idea? Like, it's really awesome. Um, so like I said, we're gonna zoom in. Boom, molecules. Because what is dopamine? It's a neurotransmitter. So it has actions in the central nervous system and it travels within the central nervous system. And it belongs to a specific class of neurotransmitters called neuromodulators. Now, those guys are really important. They can regulate the responsiveness of neurons to each other. They can change the firing rate of neurons. And they can even sort of, they can change the synaptic connections in those areas as well. I mean, that's huge. Those are huge. Being able to change how neurons respond to each other and sort of manipulate the circuitry within your brain is the basis for so many things like learning and memory. So forming memories, storing memories, all the different types of learning involve this concept of synaptic plasticity, which is the strengthening and weakening of certain connections between neurons. Uh, that is sort of the the more zoomed out things that are going on when you are doing cognitive processes, basically. So the fact that dopamine has this pretty big role in affecting those connections is mm, so good. It's so good. um, let's see. So I'm sorry. I'm just so like circuitry, synaptic plasticity. It's so fascinating to me. Anyway, dopamine. It looks kind of familiar, maybe. Maybe it looks a little bit like this guy. Maybe it looks a little bit like this guy. These three, dopamine, noradrenaline, aka norepinephrine, adrenaline, aka epinephrine, they're all monoamines. And they're all a part of the same biosynthetic pathway, which means that they get made from each other, basically. They are all made from the amino acid tyrosine. Which, fun fact, tyros means cheese in Greek, and tyrosine was first isolated from the cheese protein, probably casein, I'm assuming. So, the more you know. Anyway, what we're looking at here is a biosynthetic pathway. So, the arrows in between are that's the direction that the reaction is going, and then in between the arrows are going to be the enzymes that are bringing uh, bringing the molecules through the process, basically turning one molecule into another, and so on. So on the left, when we start with tyrosine, tyrosine gets acted on by an enzyme called tyrosine hydroxylase. What that enzyme does is it adds a where's my pen? It adds a hydroxyl group. It adds an to whoop to tyrosine right there, and it turns it into something else. It turns it into L-dopa. L-dopa is the direct precursor to dopamine. L-dopa gets acted on by aromatic amino acid decarboxylase, and it just twims. It just does a little trims off that carboxyl group, and it turns it. This is the carboxyl group. Oh my god! And it turns it into dopamine. So dopamine can continue to be metabolized and turn into noradrenaline, and then noradrenaline can get turned into adrenaline. But of course, we're gonna hang with dopamine. We like dopamine. We're gonna hang there. Who's talking about CBAT in my chat? Who's bringing up CBAT? Your moderator. You're fired. You're banned. Oh my gosh. So, as you can tell, maybe by the fact that adrenaline and noradrenaline are involved in this pathway, dopamine can be synthesized in the adrenal medulla, in your adrenal glands, and that's cool. But like I said, we really care more about the CNS. I love hanging with dopamine too. I love having it in my brain. Gretchen, you're dopamine. You makes me release dopamine. I release so much dopamine, and it signals to my brain that you're a reward, and I want to be around you. I want to approaches you. Hell yeah. Oh my god. So, in addition to being made in the adrenal medulla, the CNS is what we care about. So, dopamine is also made in dopaminergic neurons in our midbrain, which is at like the top of our brainstem. It's like right up in there. And that's, that's where we're going to be zooming in on. Also, quick note, anytime I end anything in ergic, dopaminergic, glutamatergic, GABAergic, cholinergic, whatever, that is referring to neurons that respond to or release a certain neurotransmitter. It is not confusing at all. I don't know what you're talking about. It makes perfect sense. So, now that we have our sort of dopamine recipe here, it's time to look at those signaling pathways. It's time to look at the molecular interactions that dopamine has that work together to sort of propagate these signals that eventually lead to dopamine's effects that we know and love so much. So, central to that signaling process, of course, is the receptor. A receptor is a protein that binds to a molecule, and when that happens, a cascade of events sort of happens that lead to an effect. And this is true. I mean, this sort of signaling is true in any biological living thing, any living thing. So, for example, here we have adrenaline. Adrenaline will bind its beta 2 adrenoreceptor in our hearts, and the effect is increased heart rate. Another example is vasopressin will bind to its V2 receptor in the kidneys and lead to water retention. So in its most simplest form, this is what the molecule binding to a receptor and having an effect. This is it in its most simplest form. In its most simplest form, like I said, like an over-the-surface kind of look at it, because, yeah, vasopressin, its effect is to increase water retention, but more like specifically, its effect is to activate a protein called aquaporin. And when that protein is activated, it'll go on to the plasma membrane of the cells in the kidney and force water inside of the cell. That's sort of the connection between these little molecular things that are going on and then the net effect of water retention. It is unbelievably fascinating. Oh yeah, keyword ligand. A ligand is basically anything that binds a receptor. Anything. So in this case, vasopressin is the ligand for the V2 receptor. It's a nice umbrella term that we can use for those kind of things. So, yeah, receptors are what connects function to a molecule, and it transforms signals from the ligand into effects like walking in a straight line and breathing and vomiting, and everything that we can do physiologically has a sort of reaction like that. And the existential dread is setting in already. Our enzymes, ligands. Ooh, enzymes. I can't think off the top of my head if there's a time that an enzyme is a ligand, so I'm comfortable saying no. But molecular biology is weird. Who knows? Maybe there could be a time where an enzyme is binding something as a way to continue a metabolic process. Ligands are going to be on the quiz, so remember it. You best remember it, and you better pronounce it right. That's a trick. You can pronounce things any way you want. My professors, every single one of my professors pronounced like one word differently. Ligand, ligand, ligand. So don't worry about that. Yeah, definitely put it in your notes. That is a really good question, though. But yeah, I would say more often than not, this is technically incorrect. I'm just using it to help visualize. But enzymes would be more related to a receptor because they have a substrate that binds to them. And I would say that the substrate is like a ligand that gets broken down. So actually, I'm going to go back to this. So all of these, tyrosine is a substrate for tyrosine hydroxylase, if that makes sense. So it's sort of, you've heard like the lock and key mechanism. that's the same for ligand and receptor. So that's why I'm like, that's an interesting thought, but I'm gonna say enzymes are not ligands. Interessante. Tegan You're in ligand? Okay, alright, Amy. You're about to strike the fuck out. I'm just kidding, I love you. You can know what I'm talking about. I do, because you showed me. You've corrupted me already. So this sort of bridge that I'm trying to build between these effects that we can see and these underlying mechanisms that are like absolutely gonna send me into like existential insanity. That's more what molecular biology is about. It's more about the second part of that. And it's not just about like the one molecule. It's everything that goes into a biosynthetic pathway or I don't know, any sort of hormonal signaling, anything like that. And it's very like there's a lot of disciplines that go into it. So like this little chart I thought was really nice. I identified with this little circle here of the combination between biochemistry and genetics in there. Because there is a lot of chemistry, but don't worry, we won't do chemistry, I swear. So yeah, dopamine would be freaking nothing without its receptors. So why don't we look at those? Chemistry is hard. That's why in like all of the science like degrees, they use it as like the which of my pretties will be weeded out of their, you know, dream career. Like it's always chemistry. Okay, so don't freak out because it is I ordered it this morning with my coffee and I put it in the fridge, and when I opened the fridge like 10 minutes ago, I was like, I love myself. I'm so nice to myself. Oh my god. This slide is very toxic. And right out the gate, I'm not going to talk about each of these receptors individually, but also false advertising because we're going to go into some shit. These are all the dopamine receptors. There's 6 of them. They're super awesomely named. D1, D2, D3, D4, D5, TAR1. So interesting. Luckily, they're kind of categorized. The D1-like receptors, D1 and D5, they're similar to each other. And then the D2-like receptors are all similar to each other. I believe they were categorized, I mean, kind of based on their function, but usually with stuff like this, it's based on how they look. It's based on their protein structure similarity and maybe even their gene structure similarity. And then TAR1 is just, it's TAR1 is TAR1. It's just its own thing. The number of receptors here just for dopamine, 6 for one molecule, and the really like diverse functions that they have kind of explain how a single molecule like dopamine can have such a wide variety of effects. These are the sort of thing, this is why like molecular biology is so fascinating and also so complicated. Stuff like this, where it's like one molecule, 6 receptors, billions of effects. It is the best. Time to blast those sequences. Oh, yeah. Oh my god, dude. Blast from the past. I used to, I took a bio, it wasn't like a whole course, it was just like a bioinformatics, like, come in, and this, like, really tired TA will teach you how to use, like, the NCBI, like, protein blast functions. And I was just messing around, and I was typing random, like, ATGCs into the thing. And the first one I got up when I pressed it was fucking cannabis sativa. It was great. Trace anime associate. You've ruined a means for me. I'm just kidding. Subconscious. It wasn't subconscious though. I don't know how subconscious conscious that was. I think it was right there, right up in there. All right, so I have to say the next few slides are gonna be triggering for some of you guys, because I said I wasn't gonna go into the specific, the intricacies of each receptor, but I am gonna talk about how D1 like and D2 like and TAR1. I'm gonna go into their molecular mechanisms. I'm gonna go into their signaling cascades. So we'll just take some deep breaths. We'll just like have a little sip of our and it's gonna be fine. So in these signaling cascades that I'm about to show you, you're gonna see that it's all about the shape changes. It's all about the shape changes. And what's the most common shape in molecular biology? The blob. Everything is blobs. Everything inside of you right now is just blobs. It's all blobs. And they all hit each other and they have fun with each other. And that's just kind of what they do. We do love blobs. It makes things very easy to like visualize. I love I love the evolution of the animations from like my AP bio class to like my freaking like eukaryotic cell biology class, and it's like, whoa, we've come a long way just in like that short amount of time. But my animations are priceless. So I've given you a second to kind of sit with this and just, it's okay. The blobs will guide us to victory. What you're looking at here is shot for shot. This is exactly what a D1-like receptor looks like. Exactly what it looks like. It is a G protein coupled receptor, which means that the receptor, which is the, oh my gosh, this guy right here, this is the receptor proper, is attached to a group of G proteins. The G just means GTP and GDP. That's what they named it for. G protein. Yeah, I'm definitely going to do Unite tomorrow. Hell yeah. Snake blob, exactly. So G protein coupled receptor. What it is in the name, so that's kind of nice. So right here, the receptor is not doing jack shit. There's nothing bound to it. There's no dopamine, and GDP is bound. And GDP is a molecule that kind of acts like a switch. So right now, we're in the off position. When dopamine binds, though, we see a little bit of a shape change in the receptor, and the change in shape sort of changes the chemical interactions taking place, and that forces the alpha subunit, the blue, to open its little alpha helical mouth and release the GDP. Bye. And pretty immediately, because the cell just has a shit ton of it, GTP replaces it. Now that the alpha subunit is bound to GTP, it's going to be in the on position. Again, the intricacies of like the chemical interactions, like that's chemistry. All you need to know is like on the surface, those chemical interactions and the shape changes are kind of one and the same, and it causes a change. The changes in shape change the chemistry, and then the chemistry is going to affect how the proteins interact with each other. So now that we have the GTP bound and it's in the on sort of position, the alpha subunit is going to dissociate from both the receptor and the beta gamma subunit because it's like unstable. The GTP, it's like it just makes everything unstable, so they just all dissociate from each other. Literally, it's just like bye. Gross domestic protein. Shit. What was it? Yeah, see ya boys as in see ya beta gamma, like get the hell out of here. I got work to do because I'm the alpha subunit. I'm kind of the top dog around here. Oh my god, where was I? Now that the alpha subunit is bound to the GTP and it's dissociated, it's got work to do. So what's it going to do? What's going to happen exactly? How is it going to like further this signal that the dopamine started by binding to the receptor? Well, I assume you guys know about ATP. Did you know that we could just maim ATP and turn it into cyclic AMP, which stands for uh what is it? Cyclic adenosine monophosphate. We just maim it, and it turns into a like a circular ATP, but with less phosphate groups. That's what we're gonna do. And the reason why cyclic AMP is so important is because it acts as a second messenger in this signaling cascade. Now, second messengers are important because they take dopamine was like an external signal, right? And it binds to the outside. Well, wow. the the second messenger is what works from the inside. Basically, it goes and it does like really important things within the cell that lead to, you know, what have you, genetic changes in protein activity that lead to the effect. So very important. The cyclic AMP specifically will pass along the signal in a way that oh wait, I almost skipped the next step, which is, in order to make cyclic AMP, the alpha subunit is going to bind to something called adenylyl cyclase. Love that word. And adenylyl cyclase makes cyclic AMP from ATP. So when we activate it with the alpha subunit, any ATP in the cell is just going to boom get converted to cyclic AMP. And this is what cyclic AMP works. It's going to be that circle, because I don't want to have that chemical structure all up in there. Oh my gosh. So the cyclic AMP, now that we have our second messenger and we have something that can like propagate our signal from the inside, it's going to pass it along by phosphorylating something. Now, I'm going to use that metaphor of like an on and off switch. If you add or take or subtract a phosphate group from something, it can act to turn it on or off. And this is totally not confusing at all. But sometimes putting a phosphate on activates it, and sometimes it doesn't. And sometimes taking away a phosphate group activates it, but sometimes it doesn't. But in this specific one, with dopamine, cyclic AMP is going to have more of an activating effect. And in order to do that, it needs to interact with a protein that's going to act as our phosphorylator. It's going to act as our true like activator that's going to get stuff going. So the first thing it needs to do is go to a protein called pKa. This the little 4 proteins here, the 2 on the right, that's pKa proper. The 2 on the left are the regulator proteins. So when those little blobby guys, they're all blobby guys. When the guys on the left are bound to it, that protein is not doing shit. pKa is not doing anything. Get those. Put the phosphate in the bag. Oh my god. So cyclic AMP, its really important function here is going to be to bind those regulators and release pKa, protein kinase A, from jail. Fun fact, a kinase is an enzyme that adds a phosphate to something. So it doesn't, pKa does like an unlimited crazy amount of things in like a lot of different signaling pathways. It makes me want to cry. But we'll just focus on 2 here that have to do with dopamine, obviously. So pKa, it's going to add a phosphate to something. It's going to add a phosphate to the NMDA receptor. When the NMDA receptor is phosphorylated, its activity is increased. And as a result, the NMDA receptor is pretty rampant throughout our brain. It has to do, again, with like synaptic plasticity and like probably cognition and stuff like that. So by phosphorylating it and increasing your activity of that receptor, it's gonna result in like memory formation and things related to its task. So this is an indirect, I guess direct. Is it direct? You know, it's like because there's so many molecules, it's hard to describe it as direct or indirect. But I'll say direct. This is like a direct effect of dopamine is aiding in the synaptic plasticity by increasing the activity of proteins by 1, directly phosphorylating the protein. The other thing that can happen is a pKa can just like go into the nucleus, no big deal, and phosphorylate a protein called Krebs. And uh, the phosphorylation of Krebs leads to the phosphorylation of Krebs in your DNA. It goes to your DNA. It recruits all these other proteins by a specific gene, and then it transcribes that gene, boop, and then it becomes a protein. So let me just break that down. When dopamine binds a receptor, or when something binds a receptor, this thing can happen where your cell is like, let me just make proteins now. It's so wonderful. It's so cool. It does overwhelm me. I like the stuff is because it's hard to be like, this is happening inside, just in me right now. My blueprints are being accessed and utilized in this manner, and I don't even have to think about it. I don't even have to like. It's just like your body just does this, and it's like so smart. I know it is. It's very violating, but also fascinating. I know the Krebs part. Don't worry about Krebs. Just it's a transcription factor. So those are things that increase gene transcription, which tends to lead to the creation of a protein, and that's the shit for me. That's the shit for me. Oh my god. Don't even get me started about when Crab gets phosphorylated, all right? We don't want to go there. Oh my god. Yeah, just it's the fact that these downstream effects are happening based on the original ligand that bound, which was dopamine, is like, it's craziness. All this, it's just craziness to me. So anyway, D2-like receptors are the complete fucking opposite. They're the complete opposite. Same sort of G protein. The alpha subunit is slightly different, but like that's fine. In this one, when dopamine binds and GTP goes into the mouth, it dissociates, but instead of activating adenylyl cyclase and leading to cyclic AMP creation, it'll just inhibit adenylyl cyclase instead, because it's like a slightly different alpha subunit. So when that happens, a cyclic AMP doesn't get made at all. So the signal does not get relayed, basically. So complete opposite of D1-like receptors. Oh wait, we'll hold on a second before we go to TAR1. Oh man. These sorts of G protein coupled receptors are everywhere. Lots of different like molecules, hormones, neurotransmitters utilize G protein receptors. And they are very, very fascinating. Just taking a second before we go to TAR1. Whew. I'm gonna I spiced up TAR1 a little bit in my explanation, and I sprinkled some Adderall on it. Literally, because Adderall interacts pretty famously with TAR1. So there's still going to be some blobs. There's still going to be some little tiny existential things happening. But we'll have Adderall. So it's fine. Okay. Just on the right side, we're going to start there. With the red dots. That's Adderall. Adderall can diffuse directly into a neuron. It does not need a transporter. And after it enters the neuron, it'll do two things. One thing it'll do is it'll let's start with, I'm gonna start with B first, actually. I know I said it's B, but I lied. One of the things it'll do is it'll go straight to a vesicle that has dopamine in it, and it'll bind a protein on the outside of the vesicle called VMAT2, and it'll make the vesicle just bleh, the dopamine into the neuron. Which of course the net effect of that will be to the dopamine will eventually get released from the neuron, and it'll increase the amount of dopamine in the synaptic cleft. Cool. TAR1 though, the other thing that Adderall will do is it will bind TAR1, the receptor. It's just a receptor. It's not a surface receptor like the G proteins were. It's a little different. It's a receptor that just hangs out in the neuron. Adderall will bind it, and TAR1 will then activate two different proteins, PKA, familiar, and PKC. PKA will bind DAT, the dopamine transporter, and it will cause the transporter to be taken into the cell and inactivated. No more transporter. PKC will also bind the dopamine transporter, but it can reverse the direction that the dopamine transporter works. Usually the transporter takes dopamine into the cell from the synaptic cleft so that dopamine can be probably metabolized or something. But with the reverse, it just, now we're spewing dopamine into the synaptic cleft. So both of those things, the internalizing the receptor and reversing it, keep dopamine in the synaptic cleft, which is what you want if you have dysregulated or deficient dopamine. You want to keep dopamine where it's going to be functional. SSRIs are like pretty famous. They do a similar thing where you want to keep serotonin in the synaptic cleft. Vesicle. I always spell vesicle wrong. I always spell it with two S's, and I get really embarrassed. But I think I spelled it. No, I spelled it right. Ha! It looks weird with one S to me, though. But yeah, so TAR1 is very, very, very different from the classic dopamine receptors. The main thing is it's not a surface receptor. Dopamine has to be inside the neuron in order for it to take place. Oh yeah, I should say, I know I use Adderall for fun as an example here, but dopamine also, obviously, will bind TAR1 and have the effects of the PKA and the transporter. Thank you. Yeah, TAR1 is a different, very different like class of receptor, but it still has effects that involve two protein kinases, and having those kinases phosphorylate proteins, and those proteins behave differently when they're phosphorylated. So yeah, dopamine gets maximized when this process is taking place with the amphetamines. Whew. These really minuscule processes that take place in our body, they somehow relate to the observable effects. And it physically hurts me in a good way that I love to think about that. But those effects, which we've sort of seen in these examples, we saw gene regulation with cyclic AMP going to PKA going to CREB, and increasing transcription. I have another example here that's just a little less abstract, I feel, where adrenocorticotropic hormone goes to your adrenal glands in order to stimulate them to produce cortisol. That's what ACTH does. And more specifically, It goes into when it binds the receptors on the cells of the adrenal cortex. Uh, the signaling cascade leads to increases in transcription of proteins that will aid in the creation of steroid hormones, of which cortisol is one of them. One example is cholesterol side chain cleavage enzyme, so first step in steroidogenesis, as well as STAR. There's a protein called STAR, and it gets cholesterol into the cell, which you need, or into the mitochondria, which you need to do in order to steroid. So, yeah, ACTH and increasing gene transcription as a way to increase cortisol. It's just it's overwhelmingly cool. Oh my god, I'm picturing. I know the Patrick. Oh my god. Star. I have to I'm gonna write in chat how that protein is spelled because my endocrinology professor was like, um, "Hi sushi." I didn't even. I was gonna say, "Hi sushi." Beautiful. This is how you have to spell star. And my endocrinology professor was like, if you don't spell it like that, I'm gonna take half your points away. She didn't talk like that. She was a lovely woman. But still, that's crazy. It has to be like that. It stands for like steroidogenic acute regulatory element. Yeah. Okay, so that was gene regulation. The other way that molecule relates to effect is electrochemical changes, of course. We can see that in muscle, which is the perfect example of nerve impulse and like electricity and neurotransmitter release kind of shaking hands because um do a chair spin. You got it. Hold on, I gotta back up a little bit. Okay, here we go. That's fun. Okay. As an electrical impulse goes down a nerve fiber that leads to a muscle cell, um, changes in the like the electrochemical gradient of cell membranes can cause little changes here and there. In this case, making the membrane potential, oh my god, positive or negative? I think it's positive. Making it really, 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 really positive causes neurotransmitter release, and the neurotransmitter will go onto the muscle cells, and more signaling cascades, more electrical changes will occur that will eventually lead to muscle contraction. What? Why? Who designed this? I love it. I want to kiss them. All right. Whew. On the on I want to kiss them on the mouth and I hope it electrocutes me. I hope it electrocutes me. Oh man. These I wanted to put these examples in because they're more wow. They're more tangible. We can imagine a kidney. We can imagine a muscle. When it comes to dopamine and its signaling cascades and then the resulting effects on cognition and emotion, those are more obscure. Those are more abstract. Message me, Andrew, and I'll get to it later. Yeah, I'll let me I'll check it out. Also, hi, welcome. Hope you're doing okay. Oh man. Okay, but just because they're obscure and convoluted doesn't mean that they're like really, really cool and interesting. So why don't we go to the brain a little bit? Why don't we go to the brain? Truly, yeah. Magnets. It's like who knows? Okay. So dopamine's main function that everyone pretty much knows about. It It does have to do with liking things, kind of, but it is so much more than that. It is so, like, it's more than pleasure, it's more than happiness, it's more than even motivation itself. Dopamine's function mainly has to do with something called motivational salience. It's a form of attention that drives us toward or away from something, and we assign salience values to things. So, like, me and the burger, that burger got a really, really high fucking salience value. I want that burger. Um, Burger King. It might be a Burger King burger. I just chose the picture with the one that I could delete the background the easiest from that didn't look gross. Burgerness. Oh yeah, we assign high salience value to that. Um and it can either motivational salience there's sort of two avenues. The one the one that I have for this burger is incentive salience. So that's when you assign like a positive and attractiveness value to a stimulus, and the level of attractiveness value is going to determine how likely you are to approach, how likely you are to try to obtain the reward. Um, we also have also I should say a reward can be pretty much anything. Like it could be anything. Doesn't have to be food. Doesn't have to be like a job. It could be so many different things. Um and I should say, yeah, motivational salience, it's toward or away from a thing, a person, a smell. Uh so the other end of that would be the aversive salience. So you can assign an aversive value to something, and it's gonna lead to you avoiding that thing rather than approaching it. Um, and dopamine mediates both of those. It's not just liking. Dopamine signals things that you don't like as well, and things that you don't want. And it's important for learning and decision making. And we'll kind of go over that when we go a little more into the reward cognition. But yeah, these cognitive processes are very fascinating and very important, of course. Um, I mean, what's more important than feeling motivated? I mean, motivated to work or write or wake up or live, like those are key right there. um and our brains have this built-in center for controlling our perception of desirable things and propelling us toward goals, um both mentally and physically. Motor control, motor aspect of dopamine. And dopamine also, I mean, cognitively updating us on, like, do I want to do this, is this too risky? um Risk assessment, of course, is a huge thing in this process as well. um and it's God, it's crazy. Yes. Yeah. Burger King is an aversive salience. That's fair. That's fair. I can count with one finger the times that I've eaten at Burger King. And if this is a Burger King burger, I'm sorry. I like burger, but we can pretend it's not Burger King if it is indeed Burger King. But yes, smelly people would have a really high aversive salience value, as would homework, especially homework. Fuck homework. Um, you know what's really, really critical for carrying out these behaviors of, like, obtaining rewards and meeting goals? Movement. Oh, excuse me. Physically moving toward or away from something. So dopamine's motor functions have to be addressed first and foremost, because it will introduce us to the brain structures that are involved in almost all of its roles, especially in reward. Um, oh no, you got food poisoning twice. My mom used to get food poisoning all the time there from the chicken sandwich, and it's like, Mom, stop eating it. Don't eat it. Don't go there. And I remember when I was really little, We went there. Don't know why. At this point, it was established that the chicken sandwich makes my mom sick. And my mom was saying that while we were in line, and I was there with my grandma, and my grandma was like, "Okay, cool. I'll have the chicken sandwich." She just fucking ordered it. It was hilarious. Oh my god. How do you fuck that up though? How do you fast food undercooking it? Like what the hell? That is just nutso butso. That is crazy to me. Okay. Where did I go? Yes, yes, yes. We're going into the depths of our brains here. And our minds into a world that is a world of truly just like transcendent concepts and absurd vocabulary that I can't really mock as a molecular biologist because we have the stupidest words known to man. But also neuroscience, I mean neuroanatomists have some just weird names for things, and I'm sorry in advance. It's not my fault I didn't name them. Whew. Okay. Gonna crack a little bit, get ready for this. So this is our brain. It's beautiful. And these are the anatomical structures that make up the motor and the reward system. And therefore, they contain many dopaminergic neurons. Together, they all make up the basal ganglia. Because neuroanatomists like to take structures and group them together and call them other bigger structures. It's just what they do. It's fine. I love it. It's okay. I don't mind. So we're going to take a cursory glance at the functions associated with each of these areas that are mediated by dopamine. So let me just flip us to the frontal view, get a little slicey, zoom in a little bit on that area right there. Yeah, there we go. Okay. Boom. This is the basal ganglia, but blobbier. We love blobs. Okay. I don't know what's more exhausting, molecular biology or neuroscience, but I love it. I love it a lot. Okay. Don't worry. I'm going to make this worse. I'm going to make this, now I'm going to make it worse. I can't lie. Okay. The first structure is the substantia nigra. That is, I slightly mentioned it earlier, not by name, but this is where dopamine's made, in the midbrain, which is like, it's like at the top of the brainstem. This is where, this is one of the groups of cells that makes dopamine. And its neurons project to other areas of the system, and it's mainly involved in motor function, and motor control, and like learning new motor skills. Next, we have the crown of the dopamine system, the striatum at the very top. It receives, oh, there we go. Makes dopamine, motor functions. Nice. So the striatum receives projections from the substantia nigra, and it also receives signals from the cortex. The cortex is like the frontal, how there's like the frontal lobe and like the parietal lobe and all that stuff. The cortex is like the outer more parts of your brain that are associated with like really like higher order shit, like your awareness and like thought and perception and emotion and stuff like that. It's more I call it higher order. I think that's the term, just to, I don't know if it's like a level of how abstract it is, because that's what it is for me, but higher order things. And your cortex communicates with lower order things like these structures right here in order to kind of update on like what's going on like the outside world, what's going on in my environment, what am I thinking, how am I feeling, that kind of thing. And sending those signals to these lower order, I say lower order, but I'm not being like derogatory, because these structures do the shit. Okay, I mean, you're gonna see that the cortex may be higher order, but this is the area where the real like decisions are made. So the cortex. Hold on, who's next? I don't want to do this out of order. Oh yeah, so I call the striatum like the crown of the reward system. That's just it has like so many like really important functions, and it has a lot of connections throughout different parts of your brain as a way to like facilitate communication about reward cognition. And I also have some substructures because why not? And we're gonna go into one of them later because it's really interesting. Okay. Is the cortex more a concept than a place? Are you asking about the cortex? Oh man. It is a place. It's just the functions that it has are very are very are a little more abstract. So like I'll use the prefrontal cortex as an example right now because I'm going to bring it up later. But boom, prefrontal cortex is like the very front, and that's where you have like your executive functioning and most of your like emotion processing. Um, it's just the way that it's organized is, I would say that the connections in the outer areas of your brain um, have to do with things that uh, oh my god, here is maybe a good way to put it. So these structures right here, you can kind of see on this like brain slice. They're like defined cell groups, right? We call them the thalamus and the this thing. The way that scientists measure activity there is like by the cells and how much work they're doing. And so if cells are doing a lot of work, you can tell that something is being activated. The cortex does not have many cell bodies. It's mostly white matter. It's mostly like the ends of nerves. It's mostly like the sensory ends of nerves. So you can't do the same sort of testing there. You have to do something else. So physically, it's different in that way. And again, I just think that the functions that it has are just really abstract. That's like the I might have made things worse, but that's like the best way that I can put it as a more of a cell kind of biologist versus a neuroanatomist. It is so fascinating. I used to get so lost in like articles about neuroanatomy and neurobiology and just being like the funniest thing is no one can agree on anything. People disagree on this being the basal ganglia. People are like the amygdala is in the basal ganglia, and there are people that are like the amygdala is like not in the basal ganglia. Like we can't agree on anything. It's so funny.
My body that makes me want to eat
this is the this is the basal eating complex. I'm so mad at you. I really all these years I've said basal ganglia, basal ganglia, basal ganglia, and I've never once made that connection. Ooh, another hilarious thing is so the term ganglia is usually it's supposed to be a group of neurons outside in your peripheral nervous system, and nuclei is refers to a group in your central nervous system. So it's supposed to be called the basal nuclei. And you might see it called that in other places, but for whatever reason, we just like to be wrong. We like to call it the basal ganglia. I don't know why. It's wrong. It's not right. Ganglia is not in your central nervous system. And yet here we are. Okay, I'm really going to make this worse. I'm sorry. Don't worry. It's okay though. It's don't worry. Okay. The other areas of this, they have their own connections and they act as intermediaries. And in fact, as you can see, it's only the orange in here that is dopaminergic. The other ones, the The red ones are GABAergic. GABA is an inhibitory neurotransmitter. And the green are glutamatergic, an activating. GABA inhibiting, glutamate activating. So, dopamine's not the only thing involved in movement, but we're going to see why it's really important. And it's sort of what it has to do with, like, reward and feeling good. So, before I do that, I just have to say that the point of the basal ganglia is to inhibit movement. It is to regulate movement. It is to prevent too much movement and like jerks and tics and stuff like that. Your basal ganglia puts like it. I think it's referred to as like the movement brake or something by neuro people. But I'm gonna break down the pathways and we're gonna get back to dopamine. So here's a scenario. Information coming from the motor cortex, let's say the motor cortex of the cortex, is sending signals to the striatum that there are a couple different actions that can take place. Let's say you can high five, or you can fist bump, or you can handshake. We're about to talk about action selection. You have multiple actions that you can do in a situation. I took the arrows away. Don't worry. So this is the situation you're in. This is what the cortex is relaying to your striatum. And your striatum is getting signals that the other person's hand is already raised. Medulla oblongata. I always have to say that in Sheen's voice from Jimmy Neutron. I think the medulla oblongata is pretty near the substantia nigra, actually. They're like neighbors. Okay. So, action selection time. The cortex is saying, here's what's happening.
Wow.
Here are the things that can happen. But the striatum is what's going to kind of, it's not, this is kind of meta, kind of weird, but the striatum is, it's going to make the decision, okay? So it's getting information. The hand's already raised. We're going to high five. We're going to do it. We're going to do an action, okay? So what's going to happen is the striatum is going to project to the substantia nigra. It's going to inhibit the substantia nigra, which is good in this situation because wait a minute, did I fuck my arrows up? I could have fucked my arrows up. This was not the slide to fuck up out of all the slides. Oh, no, I didn't. It goes to the substantia nigra and the globus pallidus internus. Ignore that name. It's not important. It's just a thing. Both of those things, normally, without any input, inhibit the thalamus. And in the last slide, the thalamus, you may have seen this. This thalamus goes back to the cortex to help carry out an action. So under normal circumstances, the thalamus is inhibited because you don't want to just randomly like high five for no reason. But now that it's time to high five, the striatum is inhibiting those things. And by inhibiting the inhibitions, that frees up the thalamus to send signals to the cortex for a successful high five. This is called disinhibition. When you inhibit something that inhibits, thereby releasing the inhibition and having an overall excitatory effect, disinhibition. And that's what happened here. You have Amy, you have a deficiency in your basal ganglia where you cannot stop. Just slap, 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 high five. Oh my god.
Yeah, it happens when I eat a lot of Thai food to get that basil activated.
Oh my god, Amy. When you eat too much basil. Okay. So this is the direct pathway of movement. The direct pathway facilitates movement. And while this pathway is taking place, other movement choices are being inhibited. So your desire to fist bump and handshake are being inhibited while we are doing the high five. It's a pretty good system. And I just want you to keep in mind that the basal ganglia normally inhibits. That's its job. The reason why we lifted the inhibition was because information from our cortex came to our basal ganglia and said we got to do an action. So here's where dopamine comes into this process. Completing a task, like high-fiving, we'll call this a task. You release dopamine in response to that. In response to these, being successful, you release dopamine from the substantia nigra. There's direct pathway. The orange is dopamine. I'm sorry. Neuroscience is so silly. But okay, you feel good from the successful high five. You release dopamine. This dopamine that's going from the substantia nigra to the striatum is going to act as a positive learning signal. What I mean by that is you did something right, and your body is going to respond to that by priming you to do it correct again. So how dopamine is going to do that is, excuse me. So make sure I'm getting my cell populations correctly here. In the striatum, there are 2 different populations of dopamine receptors. Dopamine is going to bind both of them and do slightly different things. So, it's going to bind D1 receptors in the striatum, and it is going to remember that the D1 receptors cause cyclic AMP release and amplification of the signal. So amplifying the signal in those areas is going to promote an action in the striatum that it already does normally. The striatum normally already inhibits the GPI. So by dopamine acting on those cell populations, it is going to ramp up that inhibition. And as you can see, by ramping up, I'll explain. I know the arrows are kind of vomit-inducing, but what dopamine is doing just in that pathway is it is releasing. Wait, see, this happens to me all the time when I read this exact thing. Okay. It is lifting inhibition off of the thalamus. Let me show you. We're just looking at this area right here. Ignore the other arrow. We're only looking at the D1 receptors. This red arrow is there normally because the basal ganglia puts a brake on things. And this arrow is also here normally. But if you have dopamine and dopamine is like, yes, super definitely inhibit the GPI right here, then the GPI is not going to inhibit the thalamus because the action of the GPI is to inhibit the thalamus. So if you inhibit the action of the GPI, it will stop doing what it's doing. And again, it's that lifting of inhibition. The other thing that dopamine does, the other arrow, is it's going to bind D2 receptors in this area here and on this pathway, which is inhibiting the GPE. Remember that D2 receptors are, they inhibit adenylyl cyclase and they don't release cyclic AMP, and so the signal gets dampened. So when dopamine binds these cell populations, it has a, did I do it? Yes, it has an inhibitory effect here. So normally these neurons going here that inhibit this are not gonna do anything, which frees up this guy to inhibit this guy. And what does this guy do? The STN, who cares? All we care about is that it activates something that normally inhibits the thalamus. So by lifting the inhibition here and allowing this to be inhibited, you don't promote this guy's, again, you're doing the same, it's doing the same thing basically. All of this is to make it so that the thalamus is free to release things to the cortex. That's what dopamine does in two of these like little roundabout, but also like weirdly fascinating ways. Dude, right? Like how it's in here, right? Like it's in here. How come I don't like understand the different like cell populations and the different layers? Like why is it so difficult? We should just be born understanding how our brains work. Okay. Neuroanatomy aside, what's going on here? As a result of you doing something successful, dopamine is priming you to do the high five again when prompted by a stimulus. You are ready to do it. This can be for like, this is especially important in reward because dopamine is going to prime you to more easily obtain a reward after you get it the first time. Truly, all the time having an identity crisis. It's like I'm squishy and I'm just fat and I eat sugar and what do I do with my life? Wait, I just kind of described myself. But yeah, arrows and drawings aside, what is happening is you are being primed to more easily obtain a reward or more easily do an action. Learning. You have just learned. This is learning. This is what happens when you learn. All of us are like squishy little fats who eat sugars and glucoses. It's the best. But yeah, this is learning. You high-fiving, that was the behavior has been enforced. That's what's happening here. I think I have the other. Oh no, I could just. Well, this is nicer to look at, so we'll keep here, but I want to say that let's say you tried to fist bump that person who was definitely high-fiving you and it was like real embarrassing. The substantia nigra just produces dopamine pretty continuously, just like all the time. But it's not enough to activate those pathways. It's not enough to activate the go pathway, the direct pathway, or even the indirect pathway that inhibits movement. So it like you're not gonna like learn. It's not gonna prime you basically. You probably will have a learning experience where it's like, okay, you're not gonna just go for the fist bump again or something like that. So as you can see, like too much dopamine in that system can lead to like tics. It can lead to tics and like impulsive movements, impulsive behavior, depending on where it is. And then not enough dopamine is gonna more be like slow reflexes and rigidity. I think that was a really, really good example of dopamine's action as a neuromodulator because, I mean, it changed the way that neurons communicated with each other within a system and made you more or less likely to perform an action based on signals coming from the cortex. Wow. What a journey. Oh man, I love this stuff. Okay. It also showed, of course, we're on the reward slide. Wow. It also demonstrated for us, I think I did kind of mention this. It wants like an ease of obtaining a reward. That's what that sort of learning process can also be applied to. In this case, the reward was like task completion and camaraderie, maybe. But yeah, so to kind of segue into reward, dopamine's actions in the motor system really function in the context of um, attempting to obtain rewards. So let's fucking talk about reward. That's the big ticket item here. Hey, Cryptic, I'm very glad. I know it got a little iffy there with the arrows. I'm so sorry. But sometimes we need a lot of arrows. Oh man. And I'm really enjoying this too. I'm so glad that you have found it informative, and I appreciate all of you a lot. I love this. um Reward. Definition right there: stimulus that we assign a positive salience value to, which motivates us to approach and obtain. I know I mentioned motivational salience earlier, so it's the drive toward or away from a person, place, thing, anything. Um, and we rewards have a function. They are necessary for things like learning and decision making. So dopamine's sort of interconnection with uh, like cortical areas, particularly the prefrontal cortex, which what I mentioned earlier, so it's right smack there. Um, the fact that it's tied to those processes shows that reward is itself a higher order process as well. Being tied to, I mean, reward itself has aspects of cognition, emotion, and learning, and all of those things shape our behaviors as a whole. They have like really, really lasting effects on behavior, um, as well as short-term effects on behavior. And we're gonna look at one particular neural pathway. But first, I need to clarify some vocabulary as it relates to reward. The most important clarification here is the difference between wanting and liking. They're completely different, and dopamine affects them completely differently. Uh, Wanting, or let's do liking first. Liking, because liking is correlated to pleasure. That's what people think about when they think about dopamine. They think about pleasure. Um, and pleasure is something that you feel when you consume a reward, not when you get a job. That's not pleasurable, necessarily, you know. But eating or like having sex, those are consumatory behaviors. Therefore, they are related to pleasure, and therefore, too, that's what liking something is. Yeah. They seem different to me, but then learning about them within the context of like uh neurochemistry, it's like, oh my god, they have completely different like pathways and like associated neurotransmitters. It's so fascinating. Hello, hamster. I'm so very glad. I am here to teach about hamburger and money. So continuing on the hamburger, uh yeah, so liking is equal to pleasure, and it's important to note that all pleasurable stimuli are rewarding. Not all rewards are pleasurable though. Um and that sort of relates to the difference between intrinsic and extrinsic rewards. Intrinsic being, you know, food and sex, things that are like give you pleasure. Um Versus extrinsic rewards, which are we're conditioned to feel rewarded by them, but they're not pleasurable. So that's going to be like money or like getting a job. Um, yeah, anything that you don't need to consume in order to feel rewarded by it. So that's going to get into the wanting. With wanting, things that you want, you don't need to, you don't consume them really, and you don't feel pleasure from them. Um, and it's more of like it's the actual quality that makes something like attractive to you, that and that drives you to approach it. Um, and ooh, there was another clarification that I was going to make. Oh yeah. So the important reason why dopamine, we can't say that it 100%, like dopamine release does not 100% correlate with pleasure, is because you get a dopamine burst when I look at my favorite drink that I want and love and want to consume. I have not consumed it yet. I'm not experiencing pleasure from the drink yet, but I'm releasing a lot of dopamine in response to seeing it because I want it. So it dopamine really, I would say overwhelmingly is more on the wanting side, although of course it does have um functions in pleasure. I mean, the hedonic hotspots in our brains, um, I believe, respond to dopamine. Um, yeah. So, a little breakdown. When we view a reward with our eyeballs, the visual information from our cortex gets sent to our reward system, um, and just by looking at something, like I said, you release a lot of dopamine, and the amount of dopamine that you release is directly proportional to how motivated you are to obtain the reward. It is directly proportional to how much you want but not like something. Um and it will, which means it's proportional to how far you will go to obtain something. If you really like money and you release a lot of dopamine, that has a very high motivational salience for you, and you'll do anything for that money. So there's a direct, it's a direct proportion kind of thing. Um, a literal reward signal. Uh, fascinating. I also want to say before I introduce or before I talk about this is I have my sources. I haven't posted them yet, but I will probably post them in the Discord. The most accessible one, they're all very dense. This guy, Wolfram Schultz, he wrote a really great paper on dopamine that is very like, it's got a lot of information, but it's so readable. And it's not like full of jargon or anything. Um, it's really good. So when I post my sources, if you are interested in like further reading, definitely this guy's paper. It's really good. And he has his own hypothesis, the Wolfram Schultz hypothesis. So he found out that if you expect a reward, if you know it's coming, you're not going to release as much dopamine as you would that as much dopamine than like the first time you got that same reward, um, or like novel rewards. Basically, you will release more dopamine. Unexpected rewards will release a crap ton of dopamine, and then if you don't get a reward that you expected, your dopamine will actually go down. Hmm. I wonder if this concept is gonna come up when I talk about a certain disease. It will. It most certainly will. But yeah, this is a like pretty good look into dopamine's connection with how we perceive rewards. So, the reward pathway. I There's less arrows, I think. What's the gambler's dilemma? I do not think I'm familiar. Oh man, I'm almost out of my Dwinky Dwink that I love. I am consuming it and gaining pleasure from it. But I can do the same thing with water. All right. So you saw the motor pathway. And just like the substantia nigra, which provided the motor pathway with dopamine, The ventral tegmental area, or VTA, provides the reward pathway with dopamine. It is in the midbrain, just like the substantia nigra. And the main area that the VTA projects to in the reward system is striatum. Specifically the ventral striatum. Specifically the nucleus accumbens of the ventral striatum. Specifically these two areas of the nucleus accumbens. It's fine. It's fine. So this is the mesolimbic pathway. Just these 2 million structures. This is the principal reward pathway, basically. And depending on which part of the nucleus accumbens is receiving the dopaminergic input, the associated functions are going to be a little different. Simplified. So the VTA projections to the nucleus accumbens shell, this is where we assign want values to rewards and reward cues, anything that might signal that there's a reward coming or a reward around. Assigning want values to both of those happens in this connection between the VTA and the nucleus accumbens shell. VTA projections to the nucleus accumbens core have more to do with motor learning within the context of obtaining a reward. And even the type of dopaminergic receptors on the neurons of the nucleus accumbens will sort of determine the type of reward cognition or motivational cognition, with D1 types being associated with the reward and D2 types being associated with aversion. Both. Again, you really, there's no like, for me, the big takeaway here is just, wow, is our brain organized? Wow, is our brain organized on the molecular level, the cellular level, the lobe level, the groove level. It's just Jeebus. And in reality, so there are, okay, so there's 2 other dopamine pathways. I don't have them here because this is the reward one. And the other, I think this one's is better characterized as well. But a lot of the pathways are just not like, I mean, these people can't even decide whether the amygdala is or isn't in the basal ganglia. I mean, there's so much controversy on like what is categorized as what. So this is the one that most people agree on, the VTA and the frickin' nucleus accumbens. But, and I mean, it's understandable because the circuitry in our brain is beyond confusing. And it's only, I mean, it's worsened by the amount of chemical messengers involved because dopamine's not the only neurotransmitter, you know? We also have glutamate, the excitatory. We have, that's cortisol? Yes. Oh, I knew that. We have glucocorticoids, which can affect your brain circuitry. We have GABA, the inhibitory boy. We have tyramine, another trace amine. And we have serotonin. There's even a lot more, but there's just, it's just so hard. But funnily enough, the best way to study these things is to study their diseased states. Whether that's neurological or psychological or both, studying diseases tends to help us understand the role of these molecules and map out the brain circuitry a bit. Just when you see things when they're screwed up, it's like a nice little comparison. So we're gonna go into some diseases now. I need a sip of water. And also serotonin, I need to point out. My dopamine necklace broke, so I have to wear serotonin. Fucking serotonin. So sad. All right, diseases. We cannot talk about dopamine without talking about Parkinson's disease. Parkinson's has to do with basically destruction of the substantia nigra. So left side, I got this nice little figure here. This right here, this black pigment, that is the substantia nigra. Nigra meaning black. That might be why they named it that. This is a normal brain. Over here, there's freaking nothing. There's the neurons of the substantia nigra are just like non-existent in this slice right here. So of course, what does the substantia nigra do? It provides dopamine to the motor system. So no substantia nigra means no dopamine in the motor system, means really like rigid movement and like usually other movement issues like tremors and stuff. And it can also, of course, I mean, you need dopamine for other things, so it can also lead to like cognitive issues as well. And like a lot of diseases, dude, there totally has to be someone. I've seen it in like any like online video game I've played. I've seen a serotonin, totally. And we will be best friends. We will high five, and we will bring people so much liking and wanting. Oh, excuse me. Like many diseases, this starts with a misfolded protein. In this case, the misfolded protein is something called alpha-synuclein. I believe it has something to do with neurotransmitter release. Yes. So, and dopamine's a neurotransmitter, so you need it for that. And when you misfold proteins, our bodies have these built-in ways of dealing with that. Because I think that can happen all the time. There's a little mistake. The protein got misfolded. We'll just attach some ubiquitins to it and throw it in the freaking trash. But if you have it happening a lot, like in Parkinson's, where it's just building up of these wrongly folded alpha-synucleins, that system is going to get way overloaded, and they're just going to become deposits. This is also Alzheimer's has a very similar pathology of the too much misfolded proteins making plaques everywhere. So in addition to just the neurons being destroyed, you have these misfolded proteins being deposited everywhere, and it's not fun. It's not the business. Oh yeah, I guess I should say that like obviously without this, no dopamine would mean that like what does the basal ganglia do? It inhibits movement. So if you don't have the regulation of the dopaminergic neurons from the substantia nigra, you have nothing to lift that inhibition, which is why it takes effort to move if you have Parkinson's. The treatment for Parkinson's is L-Dopa, which you may remember from the biosynthetic pathway of dopamine. It is the precursor to dopamine. And the reason why we have to give them L-Dopa instead of straight up dopamine is dopamine can't cross the blood-brain barrier. Plot twist. It can't leave and it can't enter. So dopamine in your brain is super separate from dopamine in your everywhere else. Okay. Another classic dopamine issue is addiction. The big player here being the nucleus accumbens rather than the motor system. Alcohol tracks dopamine. Okay, remember when I said that predicted rewards release less dopamine than novel or unexpected rewards? Well, that's kind of messed up here in addiction. That's kind of muddled. And the nucleus accumbens is usually really, really good at regulating dopaminergic responses to reward. But in a state of addiction, the nucleus accumbens kind of becomes the cool ant. In a really bad way though. Because it doesn't do as much to stop us from like continually self-administering rewards. Um, and the mechanism, it has to do with dopamine, but of course, there's another player. And we get to go back to molecular biology because there's another protein involved called delta phosphate. It's beautiful. I love her. Uh, every single addiction from heroin to shopping involves overexpression of this protein, delta phosphate. Delta phosphate is a transcription factor, which is a protein that promotes transcription of certain genes. So, it will get expressed. I'll go into like the things it upregulates, but the things uh it will get expressed in response to any drug, anything that you pathologically uh anything that you have a pathological amount of motivational salience for, whether it's cocaine or a fucking hamburger, will uh increase the expression of delta phosphate. Um uh it will, and like I said, it'll upregulate certain proteins that will lead to a state of addiction. And there are two main ways that it does that. The first being reward sensitization. Um, Normally, when you re-administer a predicted award, it doesn't produce as much dopamine as before, but the effects of delta phos B make us more sensitive to the drug. Uh, um, more sensitive to the, what's the physiological? More sensitive to the dopaminergic inputs to the nucleus accumbens, because there's nothing sort of inhibiting them. So what happens is you get an increase in the incentive salience value of the drug, rather than it staying the same or going down. Like really to a pathological degree does the incentive salience go up. And this is only the wanting, by the way. Only the wanting, not the liking. People who have addictions to like the really, really addictive drugs like heroin and meth, they continually want to do the drug and like their wanting is always increasing. But at a certain point, they don't like it. They stop liking it. It's just, it's like jail, basically. I mean, they keep wanting to do something that they don't even like, which I think, again, really shows how dopamine overwhelmingly functions in wanting things. Yeah, yeah, it's terrible. It's so bad. Uh, um, the other change is a. I know I'm kind of blocking it, so I'm gonna let me just do this for a sec. The other change is you get desensitized to aversive stimuli. So you basically care less about potentially harmful cues or punishments in regards to your pathologically salient reward. So you just really, really get locked in. Yes, it increases the dopamine and it makes you more sensitive to it. So like I will use I think I can use myself as an example. I'm forming the example. I have a problem with Nutella. This is going to sound so stupid, but it's real. The more right now, I really want it, but I'm not eating it. But if I were to have it right here and I were to eat it, I would like continually want to keep eating more. I feel sick. I feel horrible. I feel so ill, but I keep going. I keep wanting, keep wanting, keep wanting, even though I don't like eating it anymore. I feel sick. I don't like this. And that is because the dopamine that is being continually released in higher than normal amounts, uh, my nucleus accumbens is not doing its job anymore because of the delta phos B of like attenuating those responses. Thus, I am just it just keeps going. Does that help? Does that make sense? And this is real, by the way. I have a problem with Nutella. I really do. Like, I'm not trying to downplay um anyone who has really serious addictions to like hard drugs or anything like that, but it's the same pathology. It really is. It's just so yeah, and I think uh I would be curious to see if over time the dopamine does go down, but then you're still sensitized to it. So even though the level is going down, your response to it might still be like increased. That'd be interesting. I'm not entirely sure about that. All right, lastly, everyone's favorite uh everyone's favorite ADHD. We can't talk about dopamine without talking about mental illness because there are lots of mental disabilities that implicate dopamine in their pathology, notably depression and its tendency to deprive one of the feelings of wanting and liking. Well, usually wanting, actually. When you have depression, you know the things that you like, but then you're like, why do I not want these things that I like? So it's, yeah, again, the fucking wanting, man. Um, I'm gonna focus on ADHD because it's very heavily studied. People are very interested in ADHD, so there's a lot of information out there about it. Um, and yeah, it's just heavily studied. Keep in mind though that ADHD shares symptoms with like a frick ton of other like mental disorders, and like the key determiner of a mental disorder is that it ruins your life. So, um like everyone has these traits and anxieties that have to do with differing or yeah, let's say differing like circuitry and neurotransmitter amounts. Um but ADHD, this is a disease. I'm talking about a disease. It has a pathology. Uh, it has neural changes that make a person less equipped to participate in society um, than someone without the disorder. And I wanted to bring that up just so that I want to be sensitive to the fact that living with a disorder like ADHD is probably less fun and relatable and more like a pathologically like inconvenient existence, and it's all because of like different anatomy and circuitry that no one asked for. So we'll get into how dopamine in ADHD is generally described as a deficiency, but and that's not untrue, but I think that dysregulation is a little more accurate. That's a little more like it's a little more broad. um uh and because sometimes there is dopamine, but it's just not where it should be at the right time, or there's nothing that will respond to it. um so I'll discuss some of the basic symptoms with regards to dopamine as well as the significance of certain genes. Molecules. So executive functions, that's like kind of the key deficiency in ADHD, and those basically have to do with cognitive inhibition, inhibitory control, working memory, cognitive flexibility, and attentional control. In not so many words, that means being able to push away unwanted thoughts, being able to tune out distractions, being able to inhibit behavior that's inappropriate for a certain situation, um, recalling things that you heard 5 seconds ago, that's working memory, uh, and task switching, that's big. Um, Dopamine neurons project to parts of the frontal lobe. Wait a minute. Okay. Dopamine projects to parts of the frontal lobe, as you can see on the my thing. As you can see over here, this is the pathway we talked about earlier. This is the VTA, and then over here, this is the nucleus accumbens. So there are also inputs to the cortex. So there's communication with these higher cognitive functions going on, as I've mentioned before. And so the frontal lobe is a big controller of these executive functions. So a mixture of basically dysregulated amounts of dopamine, dysregulated connections between those areas, leads to not keeping track of time very well and forgetting things, interrupting people, being paralyzed by having too many things to do, becoming restless when not having enough tasks, or even like being distracted by a sound and not realizing until hours later that you were distracted and you didn't do anything. Those are the things that are just that get kind of messed up if your circuitry in those areas has insufficient neurotransmitter, or if the circuitry itself is messed up, or if the anatomy itself is different. Many people take those abilities for granted, but like people with ADHD have to struggle with those seemingly different tasks just because of those differences. I think, yeah. Ooh, sorry. A little stretch. ADHD brains. Certain brain areas are reduced in volume if you have ADHD. The big one, of course, being the striatum. So it's not in ADHD. It's not just the volume that's decreased, but it's also the receptor density, the dopamine receptor density, and like the amount of dopamine activity in that area. And this is true for other parts of the basal ganglia. They might have these issues as well, but it's mainly, I mean, the striatum it's the crown of the basal ganglia. So that is like kind of the big one. So, and because of that, anatomically speaking, the someone with ADHD, their system for putting on the brakes when it comes to unwanted like behavior and movements is impaired, because again, the basal ganglia is supposed to inhibit those things, and if there's not a lot of activity going on there, then there's not a lot of behavioral inhibition going on. So with it physically being smaller and you having less dopamine swimming around in that area, not having good receptor density, that also means that the feeling of being rewarded is really hard to come by. Imagine imagine having to chase the feeling of being rewarded. Maybe a lot of you don't really have to imagine that, and it's like, I mean, that's terrible. So that's why those with ADHD are associated with doing activities that are sort of filling those voids, finding dopamine releasing activities to do in order to, what's it called? Compensate. Oh my god. In order to compensate for their dopamine deficiencies. Like they almost only want to do dopamine related activities because of this dysregulation within their reward system. So that's why an individual with ADHD task switching from something like playing a video game, which is dopamine inducing, to doing chores is like impossible. And that's why what is it? What's that? A lot of parents are always like, my son can play Pokemon trading card game for 8 hours, but he won't do his homework for 10 minutes. It's like, yeah, because the way his brain is wired makes it hard for him to pull away from things that give him what he doesn't have in his brain. It's just terrible. I'm gonna go back to molecular biology and talk about some genes that are associated with or some genes slash proteins that are associated with ADHD. Yeah, getting some flashbacks. It's yeah. I feel like a lot of people with ADHD had to hear things growing up about like you would have worked harder if you wanted it, or like I don't know, just like other effed up things like that are like soul destroying. You wouldn't have lost that thing if you cared about it. Molecular biology. Okay. Because ADHD is so well studied, there are quite a few candidate genes that have been mentioned and studied. Yeah, sorry. Me as well. Oof. The first, of course, is the dopamine transporter, or DAT. Uh, Probably pretty obvious. I mean, I did mention the dopamine transporter when we talked about TAR1 and how Adderall interacts with it. Haha. But because its job is to take up dopamine from the synaptic cleft and bring it back into the cell to break it down, um, it seems that increased DAT activity is associated with ADHD. So, sorry. I know there's like this gene versus protein thing, and I said relevant genes, but I put a picture of the protein. That's because it's usually the gene itself being upregulated or downregulated, and what's the product of the gene? The protein. And very often changes in the DNA, so like a single nucleotide polymorphism, a one-letter change in the code, can change the shape of the protein and affect how the protein functions. So with increased DAT activity in ADHD, something about the protein, either something about the protein makes it more efficient, or something about the gene itself. It maybe the gene itself is upregulated, so there's just a bigger DAT density. Like it could be anything like that, but yeah, the issues with the dopamine transporter are implicated. Another gene is, oh my god, the D2 dopamine receptor. What? That's so weird. We know that we need that in our brains to communicate. So if you don't have as much receptor density, or if the receptor itself is weirdly shaped, it's going to decrease the activity, the dopaminergic activity, and the communication in those parts of your brain, which, remember, pretty important parts of the brain use dopamine. And that's going to lead to my example was reward deprivation, but there are, of course, decreased memory and cognition and other things like that. That could potentially be affected by if you have a messed up or just not enough D2 dopamine receptors. And yeah, I think it could be downregulation, or again, like the code is changed, and so you just have like a weirdly shaped receptor that doesn't work. One of those. Okay, last one. SNAP25. This one doesn't. It's not a dopamine thing on the surface, but SNAP25 is needed for neuronal growth and establishing synapses and stuff like that. Not just like right now in my brain as we speak, but also during development, during the very important embryonic development of your nervous system. Exactly. Oh snap! And with ADHD, it seems that there are some um, sort of differences with this protein's expression, and I think did I have specific notes? Let's see. Because I mean, it makes sense because children with ADHD are shown to have like, what is it? Like the connections that are made in their cortex they happen a lot later than people without ADHD, and there are volume differences in like white versus gray matter noticed in kids with ADHD. So my thinking, this is not like a definitive thing that I read. This is just my thinking, is that SNAP 25 would have to do with something like that because if you if there's something wrong with it and it's just like the development is slow, it's taking longer for the connections to be made, the connections aren't as quality, then that would make sense that something like this would affect it. And how it has to do with dopamine is it's needed for a neurotransmitter release, and dopamine's a neurotransmitter, so there. I figured there was some sort of connection there. Let's see. Oh yeah, and inadequate connections I think could be a potential issue with Snap 25. So I am so happy to report that you guys made it. We did it. Uh, I love dopamine, obviously. I think it's really cool. And I hope that you guys had fun being totally indoctrinated with molecular biology and neuroscience. Mostly molecular biology. It's the superior discipline. I can't wait to do this again. I am thank you guys so much for hanging out and listening. This was the best. I didn't want to do like a stream lecture for the longest time. I just preferred doing the video ones, but this was great. I had a lot of fun.